Hi guys, welcome to week 2 of Bio 221 microbiology. We finished our introduction week with our intros and profiles. Thanks for all the great discussion. It's been great to get to know some of you. I'm excited for this semester. We're starting week 2, lesson 1, so you'll see I wanted to kind of walk you through some of the steps here on a classic iLearn page, when some of the things that you should be aware of. Each folder for each lesson starts with notes from instructor. This is where I'm going to put this video, and I'll put in some other notes will come here. Every week you'll see this. I also have the disease presentation groups. I've emailed everyone about the group that you are in. I also have attached the actual file, and you can see the attachments down here on the bottom. By clicking that, you'll see the whole class and the groups you're in. Starting the disease projects is a big part of this week. You're going to want to get started as soon as possible. And so I'll talk about that in just a sec, but I wanted to remind you, under this lesson folder you can see all the different tabs. The great thing to look at is the pacing guide. The pacing guide is going to show you what we'll be covering each week and what things suggested of when you should be working on them. And so you can scroll down and see all these different activities that you're going to be doing. A clinical application. This is every week you'll be doing a clinical connection, doing some reading, some practices, and yeah, so just to walk you through, this week you'll be doing a clinical connection, looking at the connection between the restoration of the gospel and the advancements of microbiology, specifically with microscopes and the ability to see things that were previously unable to be seen. Microscopy is a fascinating field. We could have a whole class just on microscopy, and this is just going to give you a brief introduction. And so if you do the readings and practices, you can either click on it here in the pacing guide, or on the left you can see reading and practices. This will give you an outline. So these are some questions to ponder, and just as a hint for the exam that's coming up, you're going to want to make sure you know the answers to all of these questions. And the answers to these questions you should be able to find when you're reading the book with these specific sections. We've tried to highlight what sections in the book are most important by listing them here. And so don't just read the book like a textbook or like a novel, but open up and study. Have these questions that you have with you as you're going through. Answer the questions. Don't just read, but really study. Okay. There's a couple tools on here as well. You can listen to the different chapters. You can also look at animations and see. There's interactive flashcards and things to help you study. So a big part of this course is it's very independent study. You need to do the readings on your own. Answering these questions is a big part. I also wanted to point out now we have vocab and learning objectives that you need to make sure you understand. And so if you go up here, actually, you can click on one of the options over here, the course folder. Under the course folder, you'll see the syllabus, the schedule, but specifically if you go down here to the lesson objectives and vocabulary, you'll see specifically the lesson and what you have to know. And I would highlight this. This is preparing for an exam. These are really important things that you'll need to know. And so for lesson 1, you can come look at these objectives, and not just being able to know the definitions, but being able to do these objectives. Demonstrate exactly how bacteria and viruses fit into classification on life on Earth. Like I mentioned previously, the quizzes, the exams, the assessments, it's very application based. It's not going to be multiple choice, find the definition of a virus. Instead, it will be describe the difference between a virus and a bacteria, or you've isolated a gram-negative bacteria. How is this different from a virus pathogen? I mean, it's very application, so make sure that you don't just know these things, but you can actually demonstrate the understanding. And so the objectives and vocab can really help. This vocab, obviously, you need to know, but not just matching definition, but know how you would use it and apply it. Okay, so this was, again, in the course folder, lesson objectives. You can find this for the whole semester. Okay, so going back down to Unit 1 and Lesson 1, you can see here, so in the pacing guide, it has the readings and practices, which I was just talking about. Another thing that I wanted to mention is that you have your groups for your disease projects. That is due week 4 of this semester, which is a crazy week because it's also the week of your exam. So the disease projects, like I said, the sooner you get started, the sooner you can get that done, the better. So you're going to want to start communicating with your group immediately. That's why I required that you have your contact information, email, call the people in your group, get something set up, do a Google Hangout, a Skype, just so you can start making plans. If you click on, let's see, if you click on just into the course information. Oh, that was wrong. The course introduction. The course introduction has a lot of resources you can find, specifically the group disease presentation instructions. This is where you can find the information on your group disease. Here we go. Okay, so this has all the assignment. You've picked your disease instructions. You're going to want to just follow this and start preparing, okay, and start getting things ready and be able to answer these questions, be able to get some basic background information, signs and symptoms, the reservoir, the etiology, transmission, how it's treated, and really get excited. Share some fun things about these diseases. It's a huge part of your grade, and it can be really fun. I mean, it doesn't just need to be a presentation, but your group can get creative as well. You'll be using your YouTube editor YouTube video editor to do that. There's some instructions here. It's a pretty basic program. If you have any questions, let me know, but hopefully between you and your group you can get that figured out. Okay, so back to lesson, or to unit 1. So the pacing guide, you're going to want to start discussing things as your group get things rolling. A great place to discuss, I mean, like I said, Skype or Hangout, but you can also use the student lounge. And so I do want you to visit that and become familiar with that this week. You can create a thread specifically for pertussis or for rabies, a specific group, and then your group can go there and comment and start discussing through the student lounge or through email, whatever is most easy. And then at the end of the week, you're going to want to do your weekly self-assessment. This happens again every week. It's called a self-assessment. It's a quiz, and it's based on the material that you've been going over in your readings and practices. So make sure you know that material really well, and you've studied, you have your vocab and objectives figured out, and then you can click on the self-assessment, and that's due Friday at midnight. The clinical submission that you're going to be working on Monday is the connection between microscopy and the restoration. You can turn that in anytime. It's due Thursday night at midnight. And then every week with your disease projects, like I said, for the next 4 weeks you'll be working on that. We do want you to submit a review. And so if you click here, there's the disease presentation review. And this is going to have basic questions, and you're going to evaluate the progress that the group has been made, what challenges you guys have had, and just basic progress with the group. And then you'll also be doing reviews on each member of your group. Okay, so you'll be providing a specific review for the person in the group, and this allows me to know feedback, to know who's actually doing the work, who's pulling their weight, and this is confidential. It's anonymous. It's not gonna come back to the student. So please be honest and let me know who's contributing, and this will all come into factors into your grade and participation. So what you'll do is you'll click Start down here to start specific questions. So questions 1 through 3 are about your group, and so you can click and do that at any point, and then you'll do these other questions for other people in your group, and that will be due at the end of the week as well. And that will happen each week just so I can see what's happening. Okay, so you'll do your readings this week. You'll start working on your discussion, your group project. You'll prepare for your quiz. You'll turn in the microscope restoration connection clinical submission, and then do your disease review, and then also start preparing for next week. This class is going to continue to progress really rapidly. We're going to get into some heavy stuff, and so make sure you're putting the time in and studying. I wanted to re-emphasize that it's pretty much like an independent study class. I mean, you need to really be sitting down with the textbook, spending a lot of time studying. You don't have the normal lecture time that you would have in an on-campus class, and so instead of the face-to-face lecture time, you really need to be putting that time with your textbook. I mean, you should be spending easy 10 to 12 hours a week on this course, okay? So you should be spending hours each day reading and studying and preparing. And that will become obvious. The quizzes and assessments are difficult, so make sure you're keeping up with the reading and the vocab and understanding. And so let me know if you have any questions with that. I think that's a pretty good intro to this week. We're starting our actual material, working on group projects, preparing for your first actual quiz. And I'm excited about it. Like I said, there's some fascinating, interesting people in this class. I'm excited to interact with you and get to know you a little bit better. Please don't hesitate to email me, let me know any questions. Again, I have office hours Thursday morning with the Connect, and you can log in and we can chat face to face, or we can text, or we can email, anything like that. Just let me know. I'm here and I want to help. Microbiology is a fascinating field. It's exciting to see, all the discoveries that we're and things that we're understanding about the microbial life and what happens on the inside of these cells. And so it's fascinating, and I want you to be able to have fun with it. And so please don't hesitate to discuss things in the student lounge, ask questions, let's get involved, and have a great time. Thanks.